And can you see the screen?
Yes, Abhi.
Okay. So I want to start once again by reviewing the last lecture for 5 minutes. And I also want to add something to that lecture, uh, which digresses from the standard methods that are used for estimating the parameters experimentally. I think the methods that I gave you last time are, in my opinion, somewhat uh, complicated, and there are simpler ways of doing it. So, um, the last lecture started off by showing how to tackle the discrepancy between the data and the experiments, uh, and 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 I covered Hertz model, which basically took the original equation, which describes the Monod's equation, which describes growth, shown here as equation 1, and to that equation it added another equation which describes maintenance. And maintenance essentially described was basically described by a zeroth order reaction. The substrate is completely burnt up um, at a constant rate m, and it can be used actually to describe the discrepancy between um, the model and the experiments for both uh, specific substrate uptake rate, specific oxygen uptake rate, specific carbon dioxide evolution rate. You just modify the m accordingly. I mean, the entire derivation was done for substrate, but if you replace this m, actually the right thing I should have used is m sub s. We throughout the derivation describing m sub s describes the consumption of substrate. Maybe here I should write for maintenance. But you could replace this m sub s by MO2, and it would then describe the departure of the observed specific oxygen uptake rate from the model. So the moment you add this constant term, you get this expression over here, and when you use these kinetics instead of the kinetics that we used earlier, you basically find that all the uh, discrepancies can be resolved. Um, First, you find obviously by the first discrepancy disappears simply because you assumed it. Um, our assumption now is that the substrate uptake rate is not directly proportional to mu, but it is m sub s plus some term proportional to mu, which is exactly the way the data looks. Since mu is equal to d, so that discrepancy will disappear. And when you do that, as I pointed out, the final expressions you get also resolve the discrepancy between the observed biomass concentrations and the biomass concentrations predicted by the Monod model. Because now you see that the biomass concentration actually starts to decline at low D. And then I basically went on to express this result in terms that are used in the literature by telling you that this term in square brackets here is often described as this is the real formula you need to remember. And then there are many ways in which the people write it. But one way in which they write it is that this term in square brackets, which is basically called y observed. And this y observed is nothing but the ratio d over rs, where rs is the observed rs, and that is equal to sf minus s divided by x. This I had sort of pointed out. Um, then I went on to describe um, the manner in which uh, this new model's parameters are determined experimentally. And I told you that it is done by plotting 1 over y observed versus 1 over d. So uh, first I showed you how the parameters are determined mu m and ks. This actually I should have mentioned when I did the Monod model. So the Monod model has only two parameters, mu m, three parameters, mu m, ks, and ysx. And it is determined mu m and ks are determined by fitting the parameters in this way. And the yield parameter is actually straightforward in the Monod model because it comes directly out of the slope of the substrate uptake rate versus D. But in this case, it is not, well, this is the way people do it. This is the way people, when you plot 1 over ysx observed versus 1 over D, the slope gives you the maintenance coefficient and the y-intercept gives you ysx. This is the way it is typically done. But there is a simpler method. I also mentioned the problem with the Perch model and asked you to take a look at Hilbert's model. But I just want to mention that there is a simpler way to estimate the parameters, and that is, without going through these complicated diversions. The simplest way is basically to simply so here is the simple way. You can always calculate the observed specific substrate uptake rates at every D, which I pointed out to you earlier. Rs is essentially D times SF minus S divided by X. This comes straight out of the mass balance. And you simply take a look at your data. If the data, so there are two possibilities. The data can be either of the form shown in the red process, which is to say it passes straight away through the origin. If the data passes straight through the origin, then you know that the Monod type model applies, which assumes that Rs is equal to mu divided by ysx, which is equal to D divided by ysx. So the slope, simply fitting this data, the slope is equal to 1 over ysx, straightforward. So going back, let me just repeat. This thing over here tells you how to estimate mu m and ks. You remember I basically said you plot 1 over D versus 1 over S. The slope is equal to ks over mu m, and the y-intercept is equal to 1 over mu m. So the first result is shown over here. The second result is shown over here. Once you have ks and mu m, the third parameter you need to determine is simply ysx, and that will come straight from the slope of this plot. Now, sometimes you may find that actually your data looks not, the line doesn't pass through the origin, but has a distinct positive intercept like this. And instead of going through that convoluted process, here is how you can determine. This is Perch model, apply Perch model, which basically says that Rs is equal to mu over ysx plus m sub s. So that means that the y-intercept of this straight line is m sub s, and its slope is equal to 1 over ysx. So the true yield and the value of the maintenance coefficient both come out of fitting this straight line. So that this convoluted method that is there in Pert's paper and also in the literature, they plot, first they calculate ysx observed, then they take 1 over ysx observed and plot it as 1 over d, and then get, from the slope they get, you know, um, uh, from the slope they get the maintenance and the y-intercept they get 1 over ysx. This seems to me to be a rather convoluted approach. Rather than go through all that straight away, you can, from the data, get the parameters. So uh, let me repeat once again. I'll just summarize it in 2 lines. In this class of models, so parameter determination from experiments. So first is determination of the growth rate parameters. These are the parameters describing the growth rate. So these parameters are mu m and ks. These are determined by plotting 1 over d versus 1 over s. And the second one is determination of the you could say substrate uptake parameter, which could be just ysx for the monode model and ysx as well as the maintenance coefficient for the PERT model. And these you determine by plotting. In my opinion, you can plot RS versus D as I have just explained above. The slope will give you the true biomass yield and the y-intercept will give you the maintenance coefficient. Or you can use the convoluted method by PERT, which actually plots, first calculates y observed and then plots that versus 1 over d, where ysx observed is defined as d over rs, which is equal to x. Let me see. Which is equal to x divided by SF minus S. This relationship actually, so this is defined as, so this is the definition of y Sx observed. This is the definition. The definition is that y Sx observed is equal to d over Rs. You calculate this. This you know from the mass balance is nothing but x divided by SF minus S. So from looking at the observed biomass concentrations and the observed steady state concentration of the substrate, you can find out ysx observed. Then you take 1 over ysx observed and plot that against 1 over d. So it seems to me this is more complicated, but very common in the literature for reasons I have never understood. And finally, I should point out that I don't know how much exposure you have to this. Actually, these linear plots that you take by, linear plots are always good, but these days MATLAB is available, and MATLAB straight away allows you to do nonlinear regression. And what that means is that you simply provide the function to MATLAB. For example, you just um, give the value. Just for example, supposing, let's go back to, for example, determining the growth rate parameters. Now, your model says that mu equals mu m times s over ks plus s, which in a chemostat, since mu is equal to d, means that this relationship holds. And supposing you provide MATLAB with your data, which basically is, here is D, here is S, here is some D1, some S1, some D2, some S2, so on and so forth, up to Dn and Sn. This is the data that you have collected. You provide this table to MATLAB, and you provide this function to MATLAB. You tell MATLAB that D is actually this function of S. It will automatically spit out the best value of mu m and ks by doing a least squares regression. Is that, have you covered that in any course, least squares regression?
Not really, sir.
So are you familiar with, like fitting of a straight line? Have you ever fitted a straight line in Excel? Um,
Yes, sir.
in which course?
Sir, in practical courses
So
where
we did this in a physics laboratory. Like we just
So-
visited this it once.
So did they tell you what is going on there? I mean,
So not exactly.
that actually takes me far beyond the course. I mean, now I'm doing statistics, so it's a little bit. So maybe I can just give you a crude explanation of what is going on here. In least squares analysis, so let's say this is our data, RS versus D, and this is the data that you got. So at least you know how to use Excel, right? To get the straight line. I mean,
Yes, sir.
huh. So when you get, let's say, when Excel spits out this straight line, um, a line like this, what it is really doing is it is trying to find out that line. So basically, what it does is it constructs the function um, at every point. It calculates, this is your, let's say, xi, right? And uh, it basically tries to, it at every d, it tries to calculate that x, which will minimize the square xi. At every point you have an xi minus an x correspond, that is predicted by the model. You understand? So let's, no, I shouldn't say xi, I should use, let's call this, make it simple, let's call this di. So this is d1, d2, d3, d4, so on and so forth. And let's say this is your predicted value. Sorry, this is the predicted value. By predicted value, I mean the value that is predicted by the straight line. So the straight line would be called predicted, and the crosses are the observed values. So at every point, take the difference between observed value minus the predicted value, square it, and add all of them. And then basically, the algorithm says minimize the sum of these squares. So you basically find that line. See, intuitively, you can see that you can jiggle the line around. You can try different variants of this line, and that for every line you can calculate this sum of the squares. So I think this will take far too much time. um, so your model, for example, says that this is actually kind of a topic in its own right, a little. Maybe we'll cover it at some other point in time, but just open up any website on method of least squares and just see what they're doing. They're basically jiggling the line around. This is the principle of the method.
So I guess to
Until
put it plainly, we can say that this is the line which the distance of which from each point is like the sum of distances from each point is minimum for this line.
not sum of the distances, the sum of the squares of the distances.
So with like when we calculate the distance, we actually take the modulus. So like basically squaring it is just to remove the negative terms, right? So if we take the modulus, then the sum of the distances would suffice.
I mean, the reason why you take the square is if you simply take, well, these are one dimensional, right? So the in a one dimensional thing, a square doesn't come up. The square comes up if you're doing it in 2D. If I have to find the distance between 2 points in one dimension, it's simply the subtracting the coordinates. Anyway, the main thing you're doing is you're minimizing the square of uh, the distance between the prediction and the observation. And that is getting me now into statistics, which I want to avoid. But this is the general principle. And when you use MATLAB to do nonlinear regression, so maybe this will be covered in your lab. It's really unfortunate that, I mean, it's actually a reflection of pretty poor training. I mean, that somebody in second year is still wondering what least squares is, third year. Not your fault, but something terribly wrong with the curriculum. So maybe I'll cover this in the lab. This is where it belongs, not here. Uh, so in MATLAB, it does the same thing, but you can give it even nonlinear equations. Do you follow what I'm saying? You don't need to plot this versus that, get linear regressions. It is still a good idea to do that. The reason why people still do the linear regression is it is much easier to see points that are departing from your fit if you do it linearly, because the human brain can see linear trends much more easily than nonlinear trends. So when you do it with these 1 over d versus 1 over s, if points don't lie on a straight line, if there are any outliers, they will immediately become obvious to you. But then ultimately, you can just put that data into MATLAB, and it'll immediately give you the best parameters. You have to give it initial guess. You have to give MATLAB an initial guess of mu m and ks. So these initial guesses you can get from your Excel 1 over d 1 over versus 1 over s fit. It will produce some mu m and some ks. Those can be used as guesses to put into MATLAB, and that will give you the most accurate numbers. Now, why MATLAB is better than the linear fits is actually, again, part of statistics, so we'll skip it. But the main point you need to remember are these 2 points that were made over here. How are the parameters determined? Mu m and Ks are determined by plotting 1 over d versus 1 over s, and the substrate uptake parameters ysx or ysx and ms can be determined either by plotting rs versus d, which is at least to me the simplest method, or if you want to go about the convoluted method used um, in the literature, they actually plot 1 over ysx observed versus 1 over d. Either way will work. Is that part clear?
Yes, sir.
So we'll go back now to I'll start a new notebook. It's becoming difficult to. So this is really the beginning of lecture 7. I guess it's the 4th today. And the next example that I want to cover, this is the next example where the Monod model fails, and again you have to add one more equation. This is as I pointed out last time, failure of the Monod model when there are multiple growth limiting substrates. And this I started explaining last time by showing you the data. I think I found a more transparent case of the data. So this is the same principle. The dilution rate is fixed. So this really this is nuts. Not able to move this. Let's try again. Okay, so this is an experiment. First, I need to describe the experiment. Notice that the dilution rate is fixed. So this is a chemostat experiment in which this is not one experiment. This is a series of experiments in which you have fixed the flow rate, the volume of the reactor, and therefore the dilution rate. It is fixed at D equal to 0.05 per hour, which corresponds to a doubling time of almost 100 hours. And you have also fixed the flow rate, so you have fixed D and also I'll call N sub F. This is the feed concentration of ammonia, which is the nitrogen source. So ammonia is typically provided as an ammonium salt. So that is also fixed. And what you are doing is you are varying the feed concentration of the carbon source. which in this case happens to be methanol. Okay, so you have a single carbon source, methanol. You have a single nitrogen source, which is ammonium, some salt, ammonium sulfate typically, or whatever it is. And um, you operate the chemostat at a particular value. So you take a look at the data. If you take a look at this, at the bottom it is saying C/N ratio. This is basically describing the ratio of the concentration of the carbon source in the feed to the nitrogen source in the feed. Since the nitrogen source concentration is constant, this is basically describing the variation of the feed concentration of the carbon source. So as you go in the right direction, all you are doing is changing SF, changing SF while holding everything else constant, including holding N sub F constant. So you start the experiment by using a relatively low concentration of carbon in the feed, and then you measure all these parameters. So uh, this is the concentration of ammonia in the reactor. In the reactor, meaning not in the feed but inside, right? Residual concentration. You are also measuring at every point the concentration of glucose, methanol. This is the residual concentration of the carbon source, which in this case is methanol. The third thing you're measuring is the concentration of biomass. This is how much dry weight do you have per liter in the reactor. Okay. Now, there are some additional measurements given in the second panel, just below this panel, which I will get to in a minute. But first, let me describe what you observe. Now, as you increase the so these are all long experiments. I mean, each of these points is a steady state. So you you have a fixed concentration of everything, including the nitrogen source. Then you perform the experiment at one concentration of methanol in the feed, observe the residual concentrations of ammonia, biomass, methanol, and a few other things. Then, after you've got the steady state reading, again you change the concentration of methanol in the feed, maybe increase it a little bit. Then again you measure the residual concentrations of ammonia, biomass, methanol, and that's how you're getting these points. So these are all steady state data. Now what you find is that as you increase the concentration of methanol in the feed, the way to the left, I'm starting from way to the left, as you increase the concentration of methanol in the feed, the concentration of ammonia comes down. The observed concentration of ammonia in the reactor comes down, and the concentration of biomass in the reactor goes up. And this continues up until a certain point, beyond which the concentration of biomass kind of becomes constant. And as I pointed out to you last time, intuitively you know that if you're supplying way to the left, since you're supplying less concentration of methanol in the feed, the growth here is carbon limited. And way to the right, when you've supplied lots of carbon, you've provided lots of carbon, lots of methanol in the feed, it's in excess. So something else becomes limiting. And in this case, they have arranged that growth is nitrogen limited here, because you have raised the carbon concentration in the feed to phenomenally high levels now. But the transition from carbon limitation to nitrogen limitation does not occur abruptly. In between, you have what is called a transition, where actually I should write both. I write dual limited because both carbon and nitrogen are limiting, and that becomes obvious because you can see if you take a look at the graph that I have shown for ammonia, it comes down, 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 down until it hits nearly zero, right? It's basically you can't pick it up from measurement. So the graph that is labeled ammonia is coming down linearly until it hits near zero. Now, so this means basically now the cells are not able to pick up any more ammonia. Now, you would think intuitively that if they can't pick up any more ammonia, then they can't make any protein. And if they can't make any protein, then they cannot grow, right? So cells are kind of screwed. Where do they go? But the experiments show that actually biomass continues to increase, even though you're not providing any more ammonia. And the question therefore arises, why is this biomass increasing? And I should also point out that actually I should actually draw the full line. So the red line this line is showing you the concentration of methanol. The blue line is showing you the concentration of nitrogen. So the point I'm trying to make here is that there is a region which I have called the dual limited region, in which both methanol and ammonia concentrations are really, really small, which means that growth is limited both by carbon as well as nitrogen. Way to the left, it is methanol that is at a very small concentration, so we call it carbon limited. Way to the right, methanol is in excess, but ammonia is really small, so we call it nitrogen limited. But in between, both are at very small concentrations, and therefore both are limiting. That part clear? I mean, first of all, just uh, this observation, and now uh, let's take a look at the biomass. And with biomass, you see that even though the nitrogen is over, if you provide more carbon, the biomass still increases. Now, here is the intuitively sort of obvious question. If there is no nitrogen available to the cells, then why is this biomass level increasing? You know, I mean, so it seems that they have no nitrogen, but if you provide them more carbon, they're still eating up the carbon. They have no nitrogen, but they're eating up the carbon. Can anybody take a guess as to what is happening here? I've already mentioned this in class.
So basically, they're forming biopolymers, and they've reduced the production of proteins.
So basically, what they are doing here is they can't make any protein, but the biomass is enhanced essentially, as you pointed out, because they're making biopolymer. So here is the concentration of biopolymer inside the. This is the concentration of biopolymer inside the cell. So you can see that they are making no biopolymer when the carbon is in short supply, right? Carbon growth is carbon limited. But as soon as the nitrogen concentration goes down, then they can't make protein. So they don't stop growing. They basically take that carbon, because you're increasing. Keep in mind, as you go to the right, you're providing more and more carbon to the reactor. Well, they eat up this carbon, you get a higher cell density, the biomass concentration goes up. But this increase is almost entirely due to the production of biopolymer. So this is, I mean, they have labeled it as PHP, which stands for polyhydroxybutyrate, which is the type of biopolymer that is being made. So you can see that they're making biopolymer. And that biopolymer production, so basically give them more carbon, they will eat it up and make biopolymer. And this continues until you can see the second vertical line, which I'm showing in green, this vertical line. At that point, they reach the stage where they can't make any more biopolymer. You know, they've just exhausted their ability to make biopolymer. Once that happens, now if you give them still more carbon, they can't consume it. So basically, everything you give just comes out, and that is what you are seeing over here. You can see that the methanol is coming out linearly. The more methanol you give, it just comes out. So actually, it should be intuitively obvious. This is kind of how people get fat, right? I mean, um, if you keep on giving people imbalanced diets, people will eat it and simply store it as fat. But up to a point, if you give it any further, then they'll simply, whatever you give comes out, right? It doesn't go anywhere. So, uh, coming back to this point, uh, this picture, the first point I want to make is that if you consider the yield, I'm trying to now first explain where the Monod model is. As I have mentioned earlier in class, the Monod model is applicable in the C limited regime and it is also applicable in the N limited regime, but with different yield coefficients. Well, first of all, why is the yield coefficient different? The reason why the yield coefficient is different is, consider, for example, the yield coefficient yxn, okay? Sorry, uh, the yield coefficient um, yxn. How much nitrogen do you consume per unit biomass? Now, it should be, I don't know if it's intuitively obvious to you, but or maybe it's easier to visualize it's a reciprocal, which is YNX. How much biomass do you produce per unit nitrogen? So this is basically inverse. It should be pretty obvious that way on the right side, the amount of biomass that you will produce per unit nitrogen is going to be higher than the one on the left hand side, because in the nitrogen limited regime, you're actually making biomass without really using a whole lot of nitrogen, because you're making biopolymer, which is completely nitrogen free. Whereas on the left hand side, all the biomass that you're making is coupled to the production consumption of nitrogen, because it's all proteinaceous biomaterial. So the values of this particular coefficient will be different. This stoichiometric coefficient will be different in the carbon limited regime and the nitrogen limited regime. Specifically, YNX in the nitrogen limited regime will be greater than YNX in the carbon limited regime. You can make more biomass per unit nitrogen in the nitrogen limited regime than you can make in the carbon limited regime. So clearly, the stoichiometric coefficients for the Monod type model in the carbon limited regime and the nitrogen limited regime will be different. Is that part clear?
Yes, sir.
So now you have one set of equations that apply over here on the left hand side and another set of equations that apply on the right hand side. And we know that these numbers are changing smoothly from the left side. I mean, if I have one number on the right hand side and another number on the right hand side, and these numbers can only change continuously. There will be no abrupt changes. You basically know that this quantity y and x will go from a low value on the left hand side to a high value on the right hand side. So that means that in the dual limited regime, which is sitting in the middle, this particular stoichiometric coefficient is definitely changing. And why is it changing? It is changing simply due to the production of biopolymer. Because the production of biopolymer inside the cell entails the production of nitrogen-free biopolymer. That is the reason why it is changing. So any attempt to use the Monod model in the dual limited regime will fail. It has to fail because the yield is not constant. That is the characteristic property of what is happening in the dual limited regime. It is the regime in which y and x is changing from a low value to a high value. Is that part clear? Total silence. Did anybody understand what I said?
Yes, sir.
Okay. So with that basic motivation, I will just come back to. So I explained everything. I will just sort of recount it. So, for example, in the dual limited regime of the transition from C limited to N limited growth, right? Y Nx increases, right? Since the, I call it in quotes because we are developing our models by thinking of them as stoichiometric coefficients. Since this, this stoichiometric coefficient is changing, we cannot use the Monod model. We cannot use the Monod model, which assumes constant yields or constant stoichiometric coefficients. So once again, the discrepancy between, so once again, the discrepancy between the data and the model, the data and the model is resolved by introducing multiple reactions. Actually, you will just add one more reaction. Not, I use the word multiple, but now, actually, before I proceed further, I should basically mention why is this even important? I mean, why do you care that? Why do you want to? Why is this system? Who cares? I mean, biopolymer. Well, so I mean, here is the practical importance. I mean, this is actually at the heart of the biopolymer industry. So, I mean, the basic motivation should be clear to you, right? I mean, there's a huge problem with chemical plastics; they don't degrade. And if they don't degrade, then your oceans and actually, living in India, it doesn't take much. Just travel on the road and see the amount of plastic all over the place. This plastic will not degrade for decades, if at all. This biopolymer, which is basically polyhydroxybutyrate, and there are other forms, polyhydroxyalkanoate. Uh, is biodegradable and therefore of considerable importance, right? I mean, so far they have not been able to make it cost effective, but the research is going on, and it will come one day. There is no other choice. Yeah, this trend of making chemical polymers will not continue. So, having said that, the production of this biopolymer is important. The next question is, why is this experiment important? Well, this experiment is actually telling you where to operate the bioreactor, right? In a very simple way, you can see. So, let me basically point out. Um, if you take a look at this picture, uh, really simple question. Would you operate, supposing your goal was to make biopolymer, would you operate the bioreactor to the left in the carbon limited regime?
No, sir.
And why not?
So, sir, in case of the ammonia, they will not form biopolymer.
There is no biopolymer in the carbon limited regime, so you will never operate it there. What about way to the right? Would you
So
make
because
biopolymer there? Way to the right?
because in that case that is nitrogen limited, and as we can show in the graph that the in case of access methyl, they are not, again they are not creating any biopolymer.
I mean, they are, right? After all, you can see pretty high concentrations of biopolymer here.
So but that is constant now, so they are not making new biopolymer.
That's the point. So the main point is actually stated differently, that way to the right, all your methanol is getting wasted.
Yes, sir.
Whatever you give is coming out. Now, in most of these bioprocesses, which involve large scale production like biofuels, biopolymers, you don't make them in grams or a few tens of grams. You make them in several metric tons per year, hundreds of metric tons per year. In these processes, the cost of the feedstock is the most important parameter. If the cost of the feedstock is high, your process will never be economically viable. Okay? So that means that if you have a situation where your carbon source is simply going out unutilized, you're wasting your feedstock. You would never operate it in the nitrogen limited regime. So you would run this process, if you did, only in the dual limited regime. Because that is the only regime in which you are using up all the carbon, you're not wasting any of the carbon that you have, and you're producing biopolymer. Secondly, within the dual limited regime, where would you operate the process? I mean, even within the dual limited regime, there is an entire range of methanol feed concentrations in which you could operate this reactor. Where would you operate it? This is also obvious from the graph here. Anybody wants to take a shot at it? I mean, obviously, way to the right, right? This is where you like to operate it because you are getting the maximum biopolymer. Are you following what I'm saying?
Yes, sir, we are.
Okay, so so now you see why looking at this problem is important. I mean, it's not an academic exercise. So going back to what I was talking, now that you want to know, you there is a need to understand this quantitatively. Well, how do you extend the model? It is done again in a very simple way. So again, your original model was growth. So that is y xs times s plus yxn times ammonia plus YXO times oxygen goes to, here I say biomass, which is C times HAOB NC, and then plus Y XC times CO2 plus YXW times water. So this is growth. But the most important point to note here is that this is the biomass that you are making in the carbon limited regime when there is no polymer. The second reaction you introduce now is storage or biopolymer production. And here you say that maybe you have some YXS prime times S, no ammonia, plus whatever it is, oxygen or what have you, goes to, now here I put down C H A prime O B prime N C prime plus whatever. This is the composition of biopolymer. Or in our example, it was PHB. So this is PHB free biomass, and this is PHB. Okay, so this is one reaction, and this is the second reaction. And the picture we now have is that if I call this reaction 1 and I call this reaction 2, then going back to the data, basically what I'm going to say is that in the left region, this region, I have only, in this region, I have only reaction 1 taking place, which is production of PHB free biomass. In the dual limited regime, basically what happens is that reaction 1, as much of it as can happen, ends at this boundary. Beyond this boundary, whatever previous reaction 1 was taking place takes place, but the new reaction that is taking place is only reaction 2. So basically, whatever you had made using reaction 1 is done, but then additional stuff is all produced by pure production of biopolymer. So reaction 2 is kicking in here. And in the third region, reaction 2 saturates. Can't do any more. So in the left region, at the right boundary of the left region, reaction 1 saturates, right? Because if there's no nitrogen available, it has to stop. Then reaction 2 kicks in the middle region, and then allows the biopolymer to grow until reaction 2 saturates. So when you are way to the right, both reaction 1 and reaction 2 have saturated, and basically nothing is happening. Anything you add just comes out. That's all there is to it. So do you follow the the basics of what I have mentioned? That part clear?
Yes, sir.
So I don't know if I wanna go into the, maybe we'll work this through exercise. You know how to use this. So again, you have two reactions now, and you know the question would be how to write how to expand on this model. I think I'll cover that next time. But maybe I should also give to you Yet another example of this, right? Because this is a fairly important, this basic idea that that there is a region in which two things are limiting is quite important from a practical standpoint. Uh, as far as production of stuff goes. So here is another example. This one I will do without going into the literature. You should have an easy time understanding it now that you know. So another system in which this kind of a thing arises is, again, here you are changing the concentration of the carbon source, the feed concentration of the carbon source. So this is the feed concentration of carbon source. And you're performing this experiment at a fixed D, dilution rate is fixed, and fixed oxygen supply, O2 supply. Okay, the mathematical details are not important at this point. So, if you do this experiment in the same way, what you will find is that basically, as you increase the feed concentration of the carbon source at a fixed D, and you can always measure how much oxygen you have inside the reactor, and you will find that the dissolved oxygen concentration kind of comes down like this, and then stays really low. Okay, now way to the left, you can also take a look at the biomass levels. They kind of go up like this. So this is dissolved oxygen, and the green line is the biomass level. So at this point over here, your dissolved oxygen levels have gone down to really low levels. If the dissolved oxygen levels have gone down to really low levels, it means that the cells cannot do respiration. But you keep on providing more carbon source, and you find that actually the cells continue to grow, but at a somewhat lower rate, with a lower slope. So when you increase the feed concentration, X still increases, but the slope is lower than the slope to the left. Can anybody guess what is going on here? I mean, if you continue to give cells, if they don't have oxygen but are still able to grow, what do you think is happening inside from the standpoint of biochemistry?
So anaerobic respiration.
Not quite anaerobic. It isn't as if the oxygen has become zero. Basically, they start doing substrate level phosphorylation. I mean, so
Right.
the stuff just goes down to pyruvate, and they generate ATP from glycolysis. So they're still getting energy, and they're still getting biomass out of it. But it should not surprise you that if they are doing glycolysis, then there is partially oxidized stuff that is being spit out by the cell. So what you find is that in this region, you start to find, so up to this point, they're making no products. But at this point, the product kicks in. So it is this part, this is the where they're doing energy generation by the glycolytic pathway, that this stuff continues until there will come a point where they can't make any more product. So this line here, this is, I put it as P. This P can be, it all depends upon the particular cell type. Some cells make ethanol, others make butanediol, yet others may make other useful compounds. But the bottom line is not one. This might actually be multiple. I'm just put down one P here. But what is clear is that the moment glycolysis kicks in, that's when the cells start making useful stuff. And then, of course, once you get beyond a certain point, then they can't do any more glycolysis. Even that is saturated. And then this graph becomes horizontal here, and this becomes horizontal here. So on the left side, growth is carbon limited. Way to the right side, the growth has become oxygen limited. And in the middle, it is limited both by oxygen and carbon. And that's obvious from the data, because you can see that not only is the dissolved oxygen, this is DO. Dissolved oxygen is 0. Oh, I didn't show you the concentration of the carbon source. So maybe I should draw that too. So um, um, so if you take a look at the carbon concentration is very, very low, very, very low. And then suddenly at the right boundary, it just starts to come out. So this is the concentration of S, which is the carbon source. So once again, in this region, in the middle region, Monod model does not apply. And why? Well, once again, here also the yield changes, right? What is the yield that is changing over here? Well, the obvious yield to take a look at is the yield Y. Which one would it be? Y O X. How much biomass do you make? Which is actually Y XO inverse. You know, if you go back to this yield over here, and you can see that because way to the right, they are much more efficient at making biomass because they are not using a lot of oxygen and still making biomass by kicking in substrate level phosphorylation. So YOX O2 limited in the O2 limited regime will be greater than YOX in the carbon limited regime. Since the yields are different at the left and the right, and the middle region is where the yield is changing, then by definition, the middle region or the dual limited region is the region where the yields are changing, and therefore the Monod model has to fail. Right? That part clear?
Yes, sir.
So this is another example. And here also, so you would extend the model by adding another reaction. I just put it in quotes because these are not really reactions. These are, these are just models. So, and in this case, what do you think the reaction will be? If the first reaction is growth as we know it, what do you think the reaction will be? I mean, in the previous case, we added biopolymer production. What do you think you'll add here? I mean, you'll add something that represents glycolysis, right? I mean, that's essentially whatever kicks in the dual limited regime is the process you will put in. So I think I've given you the basic outline. Um, um, Now the rest I think we'll check by doing exercises. The last point is, so I have already shown you the failure of the Monod model because of changing stoichiometries. The next thing I want to point out, this is actually the failure of the Monod model. Due to lack of, lack of a better word, biochemical structure. I mean, if you have, since you have done so much biochemistry and molecular biology, the first question you have to ask is, uh, there is so much going on inside the cell, but this model does not have one word about anything. It just has one something called biomass. It has nothing about, you know, enzymes or, you know, ribosomal RNA or, you know, all this complicated stuff you study. Does that play any role? Well, it does. And there are obvious cases in which it'll break down. Here are some really simple examples. So, you know, you should also know the limitations. When you do an experiment, so this is a very simple experiment. This is simply inoculating the cells. You take the cells out of the freezer and inoculate a culture, say the shake flask contains glucose plus salts, which is typically called minimal medium or a defined medium. And when you do the experiment, you basically find, so again, you're plotting the natural log of x. And you find that the graph, you already did a numerical example, there is a lag phase followed by exponential growth and then the stationary phase. So this is the lag phase, this is the exponential growth phase, and this is the stationary growth phase. Now, think about this lag phase. Can the Monod model, will the Monod model ever predict the lag phase? So keep in mind that you have started the experiment, you took an inoculum, and you basically put it into a culture with lots of glucose. Let's say this is 5 grams per liter. Everything is in excess. Can you ever get a lag phase out of the Monod model? And if not, why not? Can you ever get a lag phase out of the Monod model?
I don't think so, sir, because it doesn't mention anything in the equations.
So that's not actually. So here is the simple answer, right? If you take a look at the Monod model, it says that for every nutrient, right? I mean, whatever it be, it has some representation like this. If all the quantities are in excess, then this is approximately equal to 1. So according to the Monod model, right from the beginning, you will have mu equal to mu m. So in the Monod model, according to the Monod model, if you did such an experiment, mu will be equal to mu m from the very beginning. From the very beginning. And the reason is that the Monod model only looks at the environment. If everything is in excess in the environment, it says we're good to go. We're ready to grow exponentially fast. But when you do such an experiment where you just took the cells out of the freezer and put them into glucose, there is internal machinery that is not ready to take up glucose. And the lag phase actually represents the production of that machinery. Actually, if you want to make the thing even more precise, replace this by lactose, which you kind of understand from your mol bio course. क्या हो गया? So this is lactose. So basically, if you take the cells and suddenly put them into lactose, then you have to make these enzymes. You have to make beta-galactosidase, you have to make the permease. These enzymes have to be made. So the lag phase, this is the period during which enzymes are made. But the Monod model basically has no, it doesn't even recognize the existence of enzymes. It simply puts a biomass and says, move on. It will never capture this, right? I mean, there's no way it will give you a lag phase. According to the Monod model, if it sees lots of substrate, you should be ready to go. The time required to prepare yourself in order to get to maximum growth rate is never going to come out because the model doesn't have it. Yeah. And one more example of the same thing is this thing that you must have studied also in your small bio course. If you take cells and expose them to glucose plus lactose, then this is observed for a long time. You basically find that the cells grow first on glucose, then there is a lag phase, and then they grow on lactose. So this is the growth on glucose. This is the growth on lactose. Has this been covered in MolBio?
Not yet, sir. Like the course is still ongoing in this semester.
So you'll cover it. Basically, what happens is that when you expose the cells to a mixture, then first they consume only glucose, and then they consume lactose. There is no consumption of lactose at the beginning, and the molecular reasons for this are sort of well understood at the level of molecular biology. It's because in the presence of lactose, in the presence of glucose, the enzymes for lactose are not made at all. Now, again, this is another system in which the Monod model will take you nowhere. So, I think that I'll call it a day now. The main point of this last two lectures was to at least give you a feeling that this is not the last word on the subject. So, this is an area in which people are continuously working to figure out how to capture these and maybe to also understand why it is necessary to capture these, right? So, I basically told you that the changing stoichiometry is extremely important for the production of biopolymers and products like ethanol or butanediol. Likewise, this phenomenon is also very important because in the real world, the cells don't grow on only glucose or only lactose. They grow on mixtures. And if they grow on mixtures, then you need to be able to represent them. So that is the front. I mean, the leading edge of the work. The Monod model, it just turns out, can capture very easily some simple systems that we work with. And so far as it works, we go along with it. So with that, I think I will put an end to this part of the growth kinetics. And tonight I'll upload the homeworks, which is about, I think, 20-30 problems, which you can solve and see where you go. Okay? All right. So I guess
Thank
I'll
you,
see
sir.
you. I'll see you
Sure.
on Monday.
Thank you, sir.
Okay. All right. Bye.